Good morning, everyone. Thank you all for joining us today for the second in our webinar series demonstrating the utility of 3D cell culture. With today's presentation on applications in cosmetic testing, our presenters will cover regulated and non-regulated testing on models including Epiderm, Ocular, Epiderm FT, and Melanoderm. And we will also have a Q&A session after the presentation, so please feel free to send us your questions throughout. And this webinar is being recorded, and you'll receive a link to access it after its conclusion. And we'll also be sending out a survey for feedback and to give you a chance to let us know what you'd like to see from MatTek in the future. It's my pleasure to introduce our presenters to you today. First is Katie Dunnick, and Katie has her PhD in Pharmaceutical and Pharmacological Sciences from West Virginia University, where she researched nanoparticle toxicity in pulmonary models. And following her PhD, she spent time researching genotoxicity and developed a unique assay to measure double-stranded breaks. She then specialized in primary cells isolation prior to joining MatTek. And also joining us today is Dr. Vicky Vega, founder and CEO of Flawless Canvas, who will be speaking about her experience using our tissues as part of product development and preclinical testing. And now I'll hand it over to Katie to begin the presentation.
Great. Thank you, Margaux, and thanks everyone for attending. Before we get into the actual webinar, I want to introduce MatTek as a company. So MatTek was founded in 1985 and began producing human tissue models in the early 1990s as a replacement for animal models. Since then, we have become the leaders in tissue model creation, and we now offer 15 models with more in research and development. We have also expanded our portfolio to include cells and media, as well as cultureware products to provide researchers with custom solutions to their complex research projects. In 2021, we were acquired by Babineau, the largest bioconvergence company, and we now have the resources of multiple sister companies to solve our customers' challenges. So to describe how these tissues are used, it's important to first understand how the tissue is made to recapitulate the in vivo environment. So we'll go over the technology and then we'll get into the webinar content. To create our 3D models, the organ tissue is received and the cells are isolated. We then expand those cells to create a large bank of the same donor. This allows us to continue using the same donor over years for consistent model production. After expansion, the cells are cryopreserved and thawed when we create the tissue. The cells are seeded onto the cell culture inserts and allowed to differentiate and stratify over time in culture. We then provide customers with a fully differentiated human tissue model that is ready to use upon arrival. As I mentioned, MatTek currently offers 15 human tissue models with more in development to meet the growing field and assist our customers with replacing animal models with human relevant 3D tissues. We offer models to recapitulate the oral cavity, ocular surfaces, pulmonary organs, skin, and intestinal and vaginal organs. Today's webinar will focus on our skin and ocular models, as these are the most commonly used for preclinical cosmetics testing. These models are created in various size cell culture inserts, depending on the model and specific research endpoints. Our epi-airway, epiderm, and epi-intestinal models can all be grown in 96-well plates to allow for high throughput screening. These 3D models can be shipped globally, either from our site in Massachusetts or from our site in Slovakia, and are shipped on a weekly basis for timely delivery. In addition, 3D models provide many advantages over animal models and 2D models. The models are all grown from primary human cells, which ensures high in vivo relevance, as demonstrated by the in vivo-like structure and function that is measured and that you'll see in our presentation today. The models are highly reproducible, which has allowed for regulatory acceptance for various toxicology endpoints and animal model replacements. The models are easy to use, and because they are all grown at the air-liquid interface, topical and basolateral exposure is possible, which allows for more in vivo relevant exposure methods. Compared to animal studies, these models are also very cost-effective. As I mentioned, our models provide an alternative for animals and have high in vivo relevance. This is important because more and more data has come out that demonstrates animal models do not translate well to humans due to differences in genetic, molecular, and cellular responses. Additionally, animal models are expensive and time-intensive, unlike MatTek 3D models. Even though animals have limited relevance to in vivo human results, more than 100 million animals are still used for testing each year in the US alone. The use of animals is not only a concern for in vivo relevance, but also for animal welfare, as these animals are typically exposed to painful compounds or inhumane conditions. For example, rabbits are used to assess the corrosivity of compounds that are characterized by ulcers, bleeding, and scabs. Luckily, 3D models, such as those provided by MatTek, are providing researchers with alternatives to animals that provide improved human relevance and do not have the same animal welfare concerns. As I've mentioned, MatTek 3D models can be used as a replacement for animal models throughout the preclinical testing phase for cosmetics. This slide does a nice job of introducing the standard or maybe typical procedure for cosmetics to make it to clinical testing and demonstrates where MatTek models can be used. After the product is conceptualized, the raw ingredient and final formulations are tested both for safety and efficacy studies. MatTek models are used heavily in both of these areas. After efficacy and safety testing, the compounds then move into clinical testing and eventually into production and launch. Now that I've given an overview of the model generation, as well as when they are used in preclinical testing, we will go into greater detail on specific studies that have utilized MatTek 3D tissues. For our webinar today, we will focus on the cosmetics field, starting with safety testing, followed by skin brightening, skin hydration, UV protection, and anti-skin aging. And then, as Margaux mentioned, Vicky Vega will join us to discuss her experiences with MatTek 3D models. As I demonstrated earlier, during cosmetics testing, safety testing is required prior to clinical studies. These safety tests can be conducted through regulated tests or through standardized tests. The regulated tests using MatTek 3D models are detailed on this slide and consist of 4 OECD testing guidelines for hazard assessment. OECD, which is the Organization for Economic Cooperation and Development, provides researchers with internationally agreed upon chemical testing methods for regulatory safety tests. For a method to receive OECD approval, numerous data points must be met to demonstrate repeatability, reliability, and accuracy. Development and approval of alternative in vitro methods can actually take greater than 10 years. The methods listed on this slide utilize either the Ocular or Epiderm MatTek models, and today, MatTek is actually the only provider of 3D tissues with acceptance in all of the listed assays with our products. On the next few slides, I will go into greater detail on the OECD eye irritation and skin irritation tests. Ocular was OECD approved in 2007 for the eye irritation test and can be used as a replacement for the Draize test that's conducted in rabbits. This model uses a reconstructed human cornea-like epithelium and can also be used for Et50 evaluations for both meat and final formulations. To assess eye irritation, following OECD testing guideline 492, tissues are received and equilibrated overnight. On day 1, the tissues are dosed for either 30 minutes for liquids or 6 hours for solids. Following the exposure period, compounds are removed and the tissue is immersed for either 12 or 25 minutes, followed by a post-treatment incubation of 2 or 18 hours. Following the post-incubation period, viability is measured via an Mtt assay. This colorimetric assay uses MTT, a tetrazolium dye, to determine viability. MTT is basically reduced by viable cells to form formazine, which has a purple color, and the relative quantity of that purple color can be measured to determine viability compared to the controls. After quantification, the viability is compared to the negative control. In this assay, it's sterile deionized water. If the viability of the epiоcular tissue is less than 60% after 2 or 18 hours for liquids or solids, this correlates with an irritant in an in vivo test. This assay also uses a positive control, methyl acetate, to ensure that the assay is working correctly. And this slide just has some representative data of what this could theoretically look like if you ran this on your compounds. In addition to regulated hazard assessment testing, during product development, eye irritation potential can also be evaluated with non-regulated methods to determine possible adverse reactions. Understanding potential reactions is important to avoid human volunteer exposure to potential irritating materials during clinical tests. These tests often include the MTT Et50 and subdraise ultramildness tests described on the top of this slide. The subdraise test is used for mild material in which the draise test is insensitive, while the MTT Et50 is used to assess the time at which a chemical causes 50% reduction in viability. On the bottom left is a study that was conducted with a compound found in eyelash serum. The bottom right study assessed a compound that was found in bubble bath products. You can see that the ET50 for the first compound was greater than 24 hours, while the ET50 in the bath product was greater than 2 hours. Based on these ET50s, both compounds were considered to be non-irritants. And this is just to demonstrate how this Et50 assay can theoretically be used with different products. In addition to the use of Ocular model for safety testing in the cosmetics field, Epiderm is approved for use in the skin irritation and skin corrosion Oecd tests. It's also used in Et50 assessments, and most recently was used to generate the extra mild test to assess mild compounds for skin irritation at Accelerate. Our Epiderm model consists of normal human-derived epidermal keratinocytes that recapitulate the human skin. Similar to the Ocular skin irritation, the Epiderm model is received on day 0 and equilibrated overnight. After the equilibration, the tissues are dosed with the compounds for 60 minutes and then thoroughly washed. Media can be collected after treatment for chemokine or cytokine analysis. However, I'll mention that this is not part of the Oecd requirement for determining irritation, but can be a nice component to add. 42 hours post-exposure on day 3, the tissue viability is measured using an Mtt assay, as I previously described. Again, the purple color that is formed can be quantified to determine viability. After quantification, the viability is compared to the negative control. In this assay, it's a DPBS. And if the viability of the epiderm tissue is less than 50% after the 42 hours, this correlates with an irritant in an in vivo test. This assay also uses a positive control, but for this assay, it's 5% SDS, and that helps ensure that the assay is working correctly. In addition to using the regulatory OECD testing guideline 439 for skin irritation, irritation potential can be tested using the Et50 assay. This assay is used to screen or used as a screen to assign expected in vivo irritancy responses based on the amount of time it takes a compound to cause an adverse reaction. It can also be utilized to rank order test formulations. Standardly, Epiderm is dosed for 2, 5, and 18 hours with compounds. However, depending on the relative reactivity of your substrate, these times can be adjusted. MTT is then used to assess viability, and an ET50 is calculated. The study that I have on the left used the ET50 assay to determine the safety of the selected dose of compound F2 over the time period of 18 hours. In this study, the positive control was predicted correctly, demonstrating that the assay worked. Researchers also tested treatment conditions to determine volumes for formulations, showing that decreased viability occurred when you increased the relative amount of the dose shown in this bottom section here. These studies then resulted in final concentrations for assessment of melanin content in a skin brightening assay, which I'll talk about in a few slides. In the past month, data was released by Accelerate showing how Epiderm can actually be used to assess irritation potential of ultra-mild compounds such as baby shampoos, baby washes, or sunscreens and lotions. Unique to this test compared to the standard ET50 test is the extension of the test timeframe from 18 hours to 24 and 48 hours. This in vitro test was validated with well-known surfactants and benchmark formulations to create a reference database. Accelerate conducted validation studies and compared the data to human in vivo patch testing and demonstrated strong correlation between in vivo and in vitro results. Data shown on this slide is representative of baby shampoos, demonstrating ET50 values ranked compared to one another. In this ET50 test, a lower ET50 value indicates a more. If you haven't seen Accelerate's work on the extra mild test, I would recommend watching their most recent webinar that provides more in-depth data on the development and validation of this assay. I'd also like to thank Carol and the Accelerate team for providing MatTek with this data to share with all of you today. So as discussed, MatTek models are used in various stages of the safety and efficacy testing of cosmetic products. After safety testing is conducted, the compounds are tested for efficacy. In today's webinar, we will discuss 4 different assays for efficacy assessment, and the first of these is skin brightening. Skin brightening efficacy studies standardly use the Melanoderm MatTek model. This model consists of human-derived epidermal keratinocytes and melanocytes that are cultured to form a multi-layered, highly differentiated model. The melanocytes undergo melanogenesis and result in tissue pigmentation that can be assessed for efficacy of skin brightening agents. Melanoderm can be made using melanocytes from Asian, Caucasian, and African American donors, depending on your desired study endpoints. The Melanoderm model helps support product development, new technology, screening of actives, finished formulations, and allows for chronic and repeated dosing exposures. To conduct the skin brightening assay, the melanoderm tissue is equilibrated for 1 hour. The test material is then applied to the apical surface of the tissue with repeat dosing. The dosing regimen is compound dependent, but it's standardly conducted daily for 14 days. The compound exposure is standardly stopped by washing the tissue with PBS, and then the tissue can be harvested for various downstream endpoints. For skin brightening, these endpoints typically involve viability for dose-finding studies, melanin concentration, histology, and macro and microscopic images. As I demonstrated when discussing ET50 values with the Epiderm model, in this study, researchers used Melanoderm, specifically Mel300B, which uses melanocytes from an African American donor, to subsequently test efficacy of their compound following the formulation optimization and safety testing. So as I showed in the previous slide, they first assessed skin irritation with the Et50 assay in the Epiderm model and resulted in the expected in vivo irritancy values. Following this irritation assessment, skin brightening was assessed by exposing the melanoderm tissues to the various formulations of the SMA 012 for 14 days, treating the tissue every other day. In this study, 2% kojic acid was used as the positive control, while deionized water was used as a negative control. Following the 14-day exposure, histology samples were collected and imaged, and data demonstrated that following the treatment, normal skin morphology was maintained. Light microscopy was then used to assess the morphology of the melanocytes, shown in this image here, and demonstrated good dendritic morphology over the length of the experiment. And finally, melanin was quantified following lysis of the cells, and absorbance was compared to a standard curve made with pure melanin. Data shown here on the right demonstrates that the formulations decrease melanin production compared to the negative control, demonstrating that the compound was not only safe in the ET50 assessment, but was also efficacious in the skin brightening assessment. In a separate study, Mel300B, which again uses the melanocytes from an African American donor, was treated with 9 different formulations to assess skin brightening. Melanoderm tissues were treated on days 0, 2, 5, 7, 9, and 12, and then assessed at day 14 once they were done culturing. They were assessed for surface reflectance and melanin production. As demonstrated in this first graph here, samples 2, 8, and 9 resulted in increased LD65 values, which is a measurement of surface reflectance, compared to the untreated and vehicle controls. The positive control, kojic acid, also showed increased values. Melanin quantification demonstrated that samples 2, 5, 8, and 9, and the positive control contain significantly less melanin than the negative control tissues, which is shown here in this center graph. Further, melanocyte morphology, shown on the right, demonstrates the efficacy of sample 2 compared to the vehicle and negative control, as well as sample 1. You can see here the brightness of sample 2 compared to sample 1, the vehicle, the negative, and the positive control. Results from this study demonstrated that samples 2, 8, and 9 were the most effective at brightening the skin. In addition to skin brightening studies, MatTek models are also used to assess skin hydration efficacy. To assess skin hydration, the Epiderm full thickness model is standardly used. This model consists of normal human epidermal keratinocytes and normal human dermal fibroblasts that are cultured to form a multi-layer model of human dermis and epidermis. The model consists of organized keratin 5 expressing basal cells, invulcrin and keratin 10 expressing spinous and granular layers, and a cornified epidermal layer. The dermal layer is composed of a collagen matrix with normal human dermal fibroblasts. To conduct the skin hydration assays, the epiderm full thickness tissue is equilibrated overnight, and then the test material is applied to the apical surface with repeat dosing if needed. Following application, the tissue can be harvested for various downstream endpoints. For skin hydration, these endpoints typically involve quantification of skin impedance, gene expression, and histological analysis. In this study, researchers evaluated skin hydration through skin impedance and gene expression changes. Epiderm full thickness was exposed to 6 different test compounds and a positive and a negative control for 30 minutes. The positive control for the study was Vaseline lotion. Skin impedance was measured prior to the apical application of the compounds, and then 0, 15, and 30 minutes following a 60-minute exposure to the different compounds. The results demonstrated increased electrical impedance for all of the compounds, but this increase began to decline 15 and 30 minutes post removal of the compounds. That's shown in this graph here. After impedance measurements, the test compounds were reapplied for 24 hours, and then the samples were lysed for gene expression analysis, shown in this graph here. UDP glucoceramide glycotransferase, or UGCG in the graph, which is responsible for de novo production of glucoceramide and associated with barrier function, was increased by all of the products, indicating that all of the products improved the barrier function of the skin. Filigrin, or FLG, in the graph, which can naturally lead to moisturizing factor and acid derivatives needed for skin hydration upon degradation, was only decreased by test article 6. Invulcrin, or IVL, in the graph, which facilitates terminal differentiation of the epidermis and formation of the skin barrier, was increased by all 6 compounds. Histology was also assessed to determine health of the skin post-exposure. The positive control test compound 1, 2, and 5 exhibit a morphology consistent with healthy tissue, such as a basal layer of the epidermis that is organized with nucleated keratinocytes and the basal and differentiated layers that are well defined. And that's represented in this top graph to show the healthy skin. Test articles 3, 4, and 6 exhibited a greater number of basal cells with condensed pycnotic nuclei and separation of the epidermis from the dermis at several regions of the tissue section, indicating cytotoxicity. And that's demonstrated in this image here. You can see the arrow pointing to the pycnotic nuclei and the separation of the cells. In general, these results indicated that test article 1, 2, and 5 could improve skin hydration without possible adverse effects. A second study assessed rebalancing moisture treatment, which was TRMT in this study, in the epiderm full thickness model compared to the leading market moisturizers, or MLM. Hydration and gene expression changes were measured after a 60-minute exposure. The results demonstrated that both the compounds increased hydration significantly compared to the untreated tissues at each time point, which is shown here on the left. Additionally, the TRMT compound had improved hydration effects compared to the leading treatment. TRMT also increased gene expression of various epidermal barrier biomarkers compared to the market leader, which is shown in the various middle graphs. Researchers also tested the two compounds in a clinical study with 2 times daily application for 2 weeks and found that the TRMT also demonstrated improved hydration in clinical studies, which helped demonstrate in vitro to in vivo correlation. I don't have that data shown on this slide, but that can be found in the reference publication. MatTek models are also used to assess UV protection of various compounds. As with skin hydration studies, UV protection studies utilize the Epiderm full thickness model also. To conduct UV protection studies, the epiderm full thickness tissue is equilibrated overnight, and then the test material is applied to the apical surface with repeat dosing as necessary for your compound. Following application, the tissue can be harvested for various downstream endpoints. For skin hydration, these endpoints typically involve cyclopyrimidine dimer formation and histological analysis to assess sunburn cell formation and overall tissue health. In this study, epiderm full thickness was treated with sunscreen, which is the product B30, and then exposed to full spectrum UV. The positive control tissue was simply just not treated with sunscreen. Following UV exposure, cyclopyrimidine dimer formation was measured and quantified. The images here are shown in the first 2 images, and then also quantified here in the middle graph. The formation or lack of formation of sunburn cells was also measured. So here, the top right image shows the positive control tissue that was exposed to UV without sunscreen, and you can see the sunburn cell formation. And the bottom is the healthy tissue that was treated with the product 830 prior to UV exposure. So this study helped demonstrate that the product 830 provided strong UV protection and reduced skin damage with pre-application. The final cosmetics efficacy assay we'll discuss today is anti-aging. And as with the last 2 applications, epiderm full thickness is also commonly used for anti-aging assessment and for efficacy testing of the cosmetic compounds. To conduct the anti-aging studies, the epiderm full thickness tissue is equilibrated overnight. Following that equilibration, the tissues are exposed to UVB and then equilibrated for 10 minutes post that exposure, and then the specific test compounds are applied. Standardly, the compounds are applied for 24 hours, but can be applied repeatedly for up to 1 week. Following this application, the tissue can be harvested for various downstream endpoints, such as changes in gene expression, cell health, and histological analysis. In this anti-aging study, epiderm full thickness was exposed to UV for 20 minutes, then either retinoic acid, or RA, or the physiologically balanced growth factors, or PBGFF, were applied for 12 or 24 hours. Growth factors were selected due to the known decrease of synthesis of growth factors during the aging process and the idea that you could potentially put those back in the skin. Following the topical exposure, the tissues were harvested and the gene expression was assessed. The results demonstrated that the physiologically balanced growth factors increased collagen 1A1 and 1A3 at improved levels compared to retinoic acid after 24 hours, which is shown here in these 2 panels. Further, FGFR receptors were induced in response to treatment, while EGFR receptors were reduced by both the growth factors and retinoic acid, shown here in the 2 center graphs. Additionally, elastin and elastin-related proteins LOXL1 and MFAP2 were induced following UV exposure, and RA, or physiologically balanced growth factor treatment, which is shown over here on the left. This study demonstrated the ability to use the 3D skin model to demonstrate efficacy of a new compound relative to the market standard RA. And this leads us into our discussion with Vicky Vega. So with that, Vicky, please take it away.
Good morning, everyone. I would like to kind of tell you the story of how we started using MatTek tissues about 10 years ago. in an attempt to find sweet spots to decrease the R&D time for product development, guaranteeing efficacy. So the main issue when we do cosmetics is, we need to develop safety and efficacious formulations, but this formulation needs to, most of the time, they are tested in clinical trials. And it's always nerve-breaking when you go into a clinical trial that, costs thousands of dollars with a formula that you don't know if that will work. You have the hint, that you have, the gut instinct that this formula will work, but you are not sure. So at that time, we were looking for basically very consistent method to screen our formulas for several months and be able to compare one to another. So one of the reasons why we didn't start with full thickness, ex vivo biopsies is because you have the differences in all the individuals and how these can basically affect your results. So when I found MatTek, it was like, kind of like the perfect model with the consistency and the reproducibility that you needed to do these experiments. So in general, like when you develop a cosmetic formula, you should kind of bucket like 6 months for the R&D process, including, product compatibility and stability in the final packaging. So anything that can basically help us to move faster in a safety and, with high level of confidence is a good model to have. So I have been using, the Epiderm system for testing potential irritation, especially when I'm using new compounds that I don't know how they react. When you try, like those well-established, cosmetic ingredients, most of the time the raw material suppliers, companies, they do the testing for irritation. But when you go higher than certain concentration, or when you have a blend of, like significantly higher percentage of active ingredients, you have the chance to get irritation. And this model is fantastic because it's fast and allow you to test several formulas at the same time, and you can move to the next phase of product development with the formulas that are less irritant and check for efficacy. The other model that we use a lot is the Epiderm full thickness model. I really love it because it's when you're lucky enough to have a PCR for 384 samples, you can run basically a incredibly large amount of samples per week on Tuesday and have the results on Friday. So Thursday or Friday, you decide which of the 15 or 20 formulas that you produce can move to the next level of a product efficacy evaluation. So things that are important to consider when you are using this model is like, although MatTek provide you with a kind of like a standard protocol, you need to optimize this for your own formulation for your own components. So be aware that this is not something that you can jump immediately and start getting results. It will require a certain level of optimization in the timing that you use to treat the tissues with your formulations, in the amount of product that you use to treat the tissues, because the whole idea is to get results related with efficacy, whatever you are looking for, that are related with the actual product penetrating through the tissue and not on the size of the tissue. So always try to avoid overflowing your samples with too much product, because you can have efficacy measure because the product is going through the cracks between the tissue and the plastic, and not because it's penetrating to the tissue. So that is one thing that is important to consider. When it comes to epidermal full thickness, and this is a request that I have been talking back and forward with MatTek for many years, it's like we need a model of aged tissue. We need, because epidermal full thickness is a model for youthful, healthy tissue. So I would love to see something that represent an aged skin model, kind of like. But because we don't have right now, the best way is to create damage. So we use UV radiation. We use also infrared radiation to measure, like specific changes associated with infrared. And there are models that can expose the tissue to pollution. So you at least have 3 different ways of produce damage in the tissue. Now, things that you can do with this is like you can apply your formulation before or after, let's say, UV radiation. So you can evaluate the protection. For example, if you're using antioxidant, you can apply before UV radiation and see the level of protection that this antioxidant will produce in the tissue. or apply it after to measure, how this antioxidant can recover faster the tissue. Things that we notice is like, when you use UV radiation, your best time point after that is 24 hours. I will not go further because the tissues are very damaged basically, and you can get fake positive and fake negative if you extend this too much. It's also important, like for your specific components if you can have a positive control. So in our case, we use retinoic acid because it's the only, FDA approved product molecule to improve the appearance of fine lines and wrinkles. So when it comes to collagen and all these things, we use basically retinoic acid to measure the efficacy of our component with comparison with something that is well established, works. The other thing that is important is like the kinetics. So it's very, especially when you use a positive control, to consider the kinetics of this. So retinoic acid, for example, start peaking in activity around 6 hours in these tissues in the model that we created. And at 24 hours, it's kind of like going down a little bit. So when you compare your different component, your different formulation, make sure that you consider different time points, because that is the reason why you use a positive control. You want to make sure that you are like comparing, your efficacy to the highest level of what retinoic acid is doing. So again, these are things that while you are doing the developing of your protocol, can help a lot to move forward with high level of confidence for a very efficacious formula. One thing that we did a lot, and I have been doing for a while, is like it's separating the epidermis from the dermis from the full thickness models. It's a fantastic method when, for example, if you're looking for collagen and you are planning to do PCR in the dermis, instead of like homogenizing the entire tissue or extracting RNA from the entire tissue, you just separate. They separate very beautifully, very easily. A couple of forceps, boom, you freeze your epidermis and you work with your dermis. You get better results, more sensitivity, because you are working basically with just the cells that express collagen. And in the future, if you are having some issue or there is an indication of a benefit that is coming from the formula that was selected, you always can go back and look at the epidermis that was, frozen, and look for gene expression and, like have extension of claims. This is basically something that was kind of like a routine when we were using the epidermal full thickness tissue. We don't discard basically epidermis, we just keep it frozen until it's further need. So one of the things that you have to consider also with the epidermal full thickness system is like your dermis is, the system is perfect for gene expression. It's not that much for visualization immunostaining, because as Katie said, this is a matrix of collagen where the fibroblasts are added. So if you're looking for the formation of collagen fiber or elastin fiber, functional ones, you will not see it there because it's lack of the full extractor. You don't have glycosaminoglycans, you don't have, other integrins or other molecules that are in the extracellular matrix that will produce that. So perfect for PCR. We are waiting for the new version of epidermal full thickness model to do, immunostaining. But with the PCR, results, you can basically select a large amount of your genes and being able to move forward with a high level of certainty to the next level or modify your formulation. One of my favorite models is the Melanoderm model, because we have the chance to have ethnicity incorporated to this model. So we have melanocytes coming from African American, Asian, or Caucasian subjects. And that is very important because, as we know, all the melanocytes produce basically different amount of melanins and different, they pack melanin different depending on the ethnicity. So if we are creating a formulation that is basically for the Asian market, you basically focus on the Asian melanocytes. My approach is always to start with the toughest one, which is the Melanoderm B, the one with African American melanocytes, because if the formula works there, it will work basically likely to work in Caucasians and of course in Asian populations. So for anyone who has done developing of skin brightening product, ethnicity is a big, like pain in your neck, because what works for type skin, if it's Patrick type 1 to 3, it will not work for 4 to 6. So this allows you to see basically to create kind of a universal formula that you can use in all the different type of populations. We use it a lot for melanin quantification, as the protocol described by MatTek. We also did Fontana mass on the staining, so we were able to actually visualize melanocytes and in the tissue. So we did basically the immunostaining, and we also did quantification of dendrite formation. That was very useful for us because we were looking for decreasing the dendrite formation, because that is how you can transfer melanin to the keratinocytes basically. So we were trying to not only decrease melanin formation, but also decrease the interaction of this melanocyte with keratinocyte, minimizing the transfer. So you can go really into mechanism of very deep into the cell biology of how your formulation works, and using this system. One of the things that is missing from this system is because it doesn't have a dermis. So all the dermal signals that basically are activating melanin production are not there. And this is very important because most of the time when you have age spots, you have a photo age dermis, a damaged dermis. So basically, we don't have those, but it's a very good predictable. We were able to do like hundreds of formulations in a matter of months, and basically in combination with the Epiderm system, we were able to say, okay, this formula is basically, it's not irritant. Remember, like with pigment brighteners, you usually get issues with irritation, and it's not irritant, and you have basically a good chances of good results in the clinical studies. We also did a lot of secreted mediator quantification by ELISA. So at the same time that we harvest tissue, we always harvest the medium, because we want to measure, like for example, pro-inflammatory markers production, like TNF alpha secretion, interleukin 8, or things like that. So that will allow us also to move forward with the evaluation of, is this a product that may create irritation or not. You can also, like we also measure secretion of hyaluronic acid into the medium. So the possibilities for measuring are endless if you are wise and save, your tissues and your soups. I think that is basically the overall view of what I have done. And this is a summary of basically the pro and the cons. I say, like produce a fast screening after reliability and efficacy, allows you to move super fast on decision when you have more than one formula. Your formulation team probably will hate you, because I was like, going back to them on Thursday, say like, okay, formula 1, 2, 3 is coming, is going to the next level, but we need to do this and this modification. So basically they were ready to go and have all the formulas ready for the next week. You have the optimization due to different, like ethnicities, and of course, like is in my experience, this is a highly predictive method to move forward to clinical trials, or if you want to do, ex vivo biopsies. The cons, as I say, like the dermal structure is limited, so good for PCR, not good for immunostaining or measuring of functional fiber formations. You need to basically be aware of this is not the skin, so it's not that you can culture these tissues for a month and and see results. You have, if you irradiate with them, probably 24 hours is your maximum time point after, and if you don't irradiate, you just treat it with with your formulations, 2 weeks is kind of like the limit time. After that, you can start getting final results due to the tissue dying basically, or not functioning well. Things that I always say to people, although gene expression is fantastic, it's always something that you need to consider, because gene expression doesn't mean protein expression. There are several other processes that happen before you get gene expression, but in general, it's a very good predictor, and you can do, as I said, measurements of things that are secreted into the tissue, or to basically, see if there is formation of, for example, hyaluronic acid. You can also measure the ELISAs on homogenates of the tissue and see if you actually have, formation of pro-collagen inside. So there are tricks that you can use to evaluate the protein formation, but be aware that gene expression is not basically translating into protein. Always consider to do optimization of your models for timing, for doses, for amount of formulation. So when we came to skin brightening, we went from what MatTek recommended to 2 microliters to treat our tissues, because our formula was extremely rich in active ingredients, so we were having issues. And even with 2 microliters, we have equal efficacy to positive control, so which is amazing. Basically, when you develop your models to evaluate efficacy, remember you can use pre and post treatment, and conclusion will be a little bit different, but will allow you to speculate a little bit further of how these, formulas are protecting or inducing repair in your tissues. I think that is it.
Fantastic. Thank you so much, Vicki. Okay, so that was a great overview of your experience with MatTek models. And as we've discussed today, MatTek models have been utilized in the cosmetics field, ranging from regulatory assays to the non-regulated efficacy assessments. In addition, researchers are using these models within their own sites. MatTek also offers in-house contract services where we work with our customers to build unique assays, which was somewhat detailed by Vicki. And again, Vicki, thank you so much for helping describe your experience with MatTek tissues.
You're welcome.
So today we demonstrated a small subset of MatTek's 3D models, including the epiderm full thickness, Ocular, and Melanoderm models for cosmetics testing. As we've discussed, these models have high in vivo correlation and can be used across numerous different applications and endpoints. In addition to the models and applications discussed today, MatTek offers a total of 15 tissue models from all areas of the body, which can be used for cosmetic, chemical, as well as pharmaceutical testing. We'll be at the upcoming Suppliers Day if you'd like to meet with any of the MatTek colleagues. Also, we'll have hands-on training, in vitro training at our Slovakia site for anyone interested in getting more experience using our models. And then this is the World Recognized Non-Animal Testing Week, so just something to keep in mind. Please reach out for more information on any of these topics. And we thank you for your interest in our webinar today. We look forward to answering your questions, and Vicki will also be part of our question panel. So if you have any questions specifically for her, please feel free to ask those.
Thank you so much to Katie and Vicki for all of the information in this presentation. And again, thank you to Vicki for joining us and sharing your experience with using our tissues and our products. We love to hear how our customers are successful in using the products that we make at MatTek. So we can get into some questions now. Feel free to continue to post questions, and we'll get some answers to everybody. So the first question actually might be a good question for Vicki, because you touched on this on your slides. The question is, can you expose to any other light besides UV, blue light, visible light, and IR?
Yes, so you guys have the model established for the UV radiation, which is great. We did infrared. That required a lot of optimization, because at that time when we did it, it was like totally new. The main thing that you have to consider when you do radiation of the tissues with a lamp source is that you are actually radiating them with the wavelength that is that you are thinking of. So there are a lot of standardization for filters and measurements of other contaminant radiations that can happen, that you need to basically address. It's totally fine if you have, something that is radiating 90% UV radiation and 5%, 90% infrared and 5% UV. Just be aware that some of the changes can be linked to the other radiation. But yes, you can do it. You can be very innovative with when it comes to different methods of damaging the tissue. Just you need to do your standardization and due diligence to make sure that you are irradiating with what you think you are.
Thank you for that, Vicki. Next question is probably a good one for Katie. How can we culture skin microbiota on the model, and which model should we use?
So we obviously don't offer the skin microbiota, but that's something if you have, you could add it. And I would say the model of interest is really going to depend on what your endpoints are. So do you need the fibroblasts in that model, or are you looking at different endpoints that maybe don't require the fibroblasts? And our technical specialists are a great resource for helping decide all of those endpoints. And we also have a lot of good technical references that we can use to assist you with that.
Thank you. Next question could be either for Katie, but might be a good one for Vicki. How to decide the dose of a cosmetic for the experiment on the model?
Okay, so what we did, we used the Epiderm system, the one that you use for irritation, and we measured toxicity basically. So we started with the 50 microliters that was suggested, and we went, in one study we use 50, 25, 12.5, and therefore like doing serial dilutions. And we observed that, like around, like let's say 5 microliters, you have no toxicity. So that was the way that we did it. The reason why we always do that when we develop our formulas is because our formulas tend to have large percentage of active. So we are talking about, like 30–40% of active ingredients. So, but using Epiderm system, do serial dilution of your formulation and measure MTT, basically. That is the easy way.
Thank you, Vicki. Question for Katie. Is the skin hydration assay validated, and what is the positive control used for it?
Great question. So it's not a validated assay. So the positive control is really dependent on your study. So one of the studies we showed today used Vaseline as the positive control. Another study used a market-leading moisturizer cream for their positive control. So it really depends on what you want to compare it to. In terms of validation, it's not a validated assay, but we always want to work with people to kind of use it as a fill-in for preclinical testing. So if you have regulators you can work with, then we can help you work with the FDA to see how you can use it in that type of manner.
Okay, is it possible to test a formulation, for example, a cream, directly on top of the tissue, or does the whole tissue need to be processed?
So we did apply what we consider, products ready to go to commercialization. So the formulas that I always test are formulas that contains everything that will go in the final product, including preservatives. So I think that is the question related to, like, you don't need to have a special blend of ingredient and later on formulate it. I will suggest you to go straight to the formula with the texture and everything that you will like to commercialize to test it, so you are not doing double work. Just be mindful of ingredients that can be metabolized in the cytochrome P450, because those create oxidative stress and can damage, can kill your tissue, but it's not that they will kill your skin, basically. So there are some moments that you need to remove certain ingredient because it's causing toxicity in the tissue, which translate into irritation. So you can do those small changes, but you should be able to try everything that will go in the final product on the top of the tissue. That is one of the beauty of this tissue.
Thanks, Vicki. Next question is for Katie. How about gamma irradiation? Are the Epiderm models suitable for that, or would the FT be a better option?
Again, this is really going to depend on what your endpoint is. So for the anti-aging, we recommend the full thickness because we're using UVB and the fibroblasts are kind of the signaling cells. So for gamma radiation, are you requiring the fibroblasts for whatever your endpoints are? And again, our technical specialists are really good at helping with these specific studies and figuring out which model suits your needs.
Okay, the next question is, can I UV the melanoderm tissues for skin pigmentation?
So no, the answer is you cannot UV these to cause the Melanoderm model to cause skin pigmentation.
And I have to add to that that although you guys gave us that answer, we tried and it didn't work. So you are correct.
Okay, another question. When would you, this is probably a good one for Katie, when would you elect to use the SIT or EIT versus an ET50?
Yeah, good question. So the skin irritation and eye irritation are both regulated assays that can be used as replacements for animals. ET50 is more commonly used for in vivo correlation, and we recommend this be used with compounds that maybe don't fit in the regulatory assay timeframes. For example, a makeup product might not be realistic to test in a regulated assay due to the volume and timeframes that are required. So the ET50 is more optimal to test for adverse reactions and kind of determine the safety of your compound.
Another one in that same vein around decision making is, when do you use Epiderm versus Epiderm FT?
Yep, another good question. So this is, as many of the questions, assay and endpoint dependent. So if you require the fibroblasts for your endpoints, such as the skin aging assay that we showed today, we recommend the full thickness model. But again, the technical specialist can really help you pinpoint which model you need to use and if you need the full thickness or the standard model.
Okay, another a good question. Do you have matching keratinocytes and fibroblasts?
Yes. So our current Epiderm full thickness standard donor is not matched, but we do have matched donors that can be combined to have the matched tissue if that's something that you need for your studies.
Okay. And then another good question for Katie. What are the lead times for the models?
Yeah, great question. So it's model dependent. Typically they vary from about 3 to 6 weeks, depending on which model you need.
And we had a couple questions about topical application of compounds in final formulations, and sort of an addendum to that. Another question is, what is the surface area of the tissues?
Yeah, good question. So it's model dependent and insert size dependent. So I mentioned we have inserts ranging from 96 well to 6 well. So our surface areas range from 0.11 centimeters squared all the way to 4.2 centimeters squared. So a lot of variation depending on if you want to do more high throughput screening or if you're looking to do protein assessment or gene analysis, you might need more tissue for those.
We had another question, if there's been any advancement of the in vitro photoallergy method.
So I am not overly familiar with advancement of that particular model. That's something I can follow up with our R&D team and we can get back in contact with you on that answer.
Okay. And then good one for Katie, what is the possibility of repeat dosing?
Super possible. We see researchers do this often, and it's really helpful to mimic in vivo exposures. So I showed a few data points today where they compared it to in vivo, where they were doing 2 times daily, and in the MatTek models, they were doing daily or 2 times daily exposures for weeks or for at least a week. And that's really, really possible depending on the model of interest.
Okay. And getting to the end of submitted questions, another good one. Do you offer cosmetic testing services such as stability and sterility testing?
So no, we don't currently offer stability and sterility testing. We offer safety and efficacy testing as an internal service.
And then one final question from an audience member. Can you assess for ROS, reactive oxygen species?
Yes, of course. So reactive oxygen species can be measured, depending on what you want to dose with. Are you looking at, is your compound decreasing reactive oxygen species? There's multiple different ways to measure that. Again, our technical specialists could provide you with publications that have done that in our models, and we'd be able to help you kind of narrow down how to test for the ROS.
Yeah, if I may add, we use, we measure lipid peroxidation due to, like T-bars. We measure glutathione levels in the different layers. We measure total antioxidant capacity, and as well as changes in size of the mitochondria using, different methods of detection for free radicals. So everything that you can do in cell cultures, you can do it in these models.
Great, thank you so much for those answers. A couple other questions that came in that I provided an answer to for everyone in the audience. One was, do you offer in-person training, and do we offer contract testing for these assays? And yes, we offer training as well as in-house testing services. You can always get in touch with us to inquire about options and costs for both of those. Another question was, can we share the slides with participants? And we will be sending a recording of the webinar. If there's any particular data that you'd like to take a closer look at, just send us a message and we'll get back to you, and we'd be happy to go over something in more detail. And we will also, we had another question about sending out articles from some of the that were referenced on some of the slides, and yes, we can send all attendees a companion packet of all of the articles and papers referenced in the presentation. So if there are no other questions from the audience, I think we'll wrap it up today. Thank you so much to everyone for joining us, and thank you so much to our presenters, to Katie and Vicki, for your time today. And we hope you all found this informative. And keep an eye out for our survey and for the link to the recording. Thank you so much for joining us.